Greetings and welcome to this XFOR Pharmaceuticals call and webcast. At this time, all participants are in a listen-only mode. A question and answer session will follow the formal presentation. As a reminder, this conference call is being recorded. It is now my pleasure to introduce your host, Dan Ferry from LifeSci Advisors. Please begin.
Thank you, operator, and good morning, everyone. Thank you for joining us today for this special earnings call, during which XFOR will focus on providing a business update and, importantly, data and results for its now completed Phase 2 study of mavorixafor in chronic neutropenia. As a reminder, on today's call, the company will be making forward-looking statements regarding regulatory and product development and commercialization plans, as well as research activities and financial projections. These statements are subject to risks and uncertainties that may cause actual results to differ from those forecasted. Description of these risks can be found in XFOR's most recent filings with the SEC, including this year's Form 10-K, which was filed on March 21, 2024, and in the company's Form 10-Q for the third quarter, which is expected to be filed shortly. Presenting on today's call will be Dr. Paula Ragan, XFOR's President and CEO, and the company's Chief Medical Officer, Dr. Christoph Arbet-Engels. I'll now turn it over to Paula Ragan to run through today's agenda. Paula?
Thank you, Dan, and thank you all for joining us. Today, we will provide an update on our operational achievements, including activities supporting the launch of XOLREMDI in the U.S. and the substantial progress to date with our ongoing Phase 3 clinical trial of mavorixafor in people with chronic neutropenia, or CN. In addition, we're excited to present the positive data from our recently completed Phase 2 study of mavorixafor in CN. These results include the final mavorixafor monotherapy data, which remain very encouraging and consistent with the data we presented in June from the interim Phase 2 analysis. We will also be presenting new data from the study, results showing that most of the participants who were treated with mavorixafor and G-CSF ended up having their G-CSF dose adjusted downward during the study, while the mean neutrophil counts for this group remained in the normal range. These data provide a first glimpse into how mavorixafor could be used in the real world if ultimately approved in CN. Equally exciting are the new insights we've gained from assessing the functionality of circulating neutrophils in a subset of the Phase 2 study participants, as well as healthy donors for a comparison. And we'll briefly review this updated safety data from the study as well. We'll then conclude and open it up for your questions, when we will be joined by our Chief Commercial Officer, Mark Baldry, our Chief Financial Officer, Adam Mustafa, our Chief Operating Officer, Dr. Mary DiBiase, our Chief Scientific Officer, Dr. Art Taveras, and our Head of Corporate and Patient Affairs, Jose Juves. So let's quickly run through the operational update, as we know you're all eager to hear about the CN Phase 2 study results and analysis. As you know, we received U.S. FDA approval in late April for our first product, mavorixafor, branded XOLREMDI, for the treatment of WHIM syndrome, a rare primary immunodeficiency disease. As with ultra-rare disease launches, our initial commercial strategy has been focused on disease awareness to support patient identification and diagnosis. While we do recognize that sales appear to be flat quarter over quarter, we want to remind everyone that some of the sales in the second quarter were product stocking with our specialty pharma, as is typical during initial launch. I'm pleased to report this past quarter we hit our most important launch goal, having engaged with all 3,400 of the initial immunologists and hematologists targeted for the first phase of our launch. And these visits and digital engagements are bearing fruit, as you can see here. Recently completed market research comparing pre- and post-launch metrics indicate that knowledge of WHIM syndrome in these likely XOLREMDI prescribers has increased and is now at more than 75%. In addition, an increasing number of physicians are screening their patients for WHIM, and 80% of HCPs surveyed report that they would consider prescribing XOLREMDI for their WHIM patients. Over the past 6 months, we've also stepped up our conference attendance, launched a peer-to-peer speaker program, and initiated a patient-focused campaign, which includes a website, new materials, and an HCP discussion guide. Reimbursement and access continue to go well, with favorable policy decisions resulting in more than 150 million lives now covered. So very good progress there. Importantly, the activities we are conducting in WHIM are yielding insights that may support our efforts to advance XFOR into a much broader indication of chronic neutropenia. And while our commercial efforts are exclusively focused on WHIM today, we do see a significant overlap between the WHIM and CN communities. Based on our claims analysis and experience in the field, we observe that a majority of hematologists that we're calling up for WHIM also see chronic neutropenia patients. And of course, there's overlap within the patient advocacy communities we engage with as well. If XFOR is approved for use in CN, we certainly expect to be able to leverage our commercial infrastructure and relationships within the WHIM community to support a potential launch into chronic neutropenia. As you all know, success in CN will be dependent on the results of our global pivotal phase 3 trial, which is progressing well, and currently participants are being screened and dosed across multiple countries. Our forward study, as we've named it, is a global trial that will engage patients and physicians across 20 to 25 countries. We can report today that we have already received health authority authorizations to initiate the trial in approximately 85% of these countries. We can also now report that approximately 40% of our planned sites worldwide are initiated, and we expect to have the majority of sites initiated in early 25. This trial is expected to enroll 150 participants at 90 to 110 sites, with a planned enrollment of 1 to 2 participants per site, which is typical in a rare disease clinical trial. Most importantly, we are on track and continue to expect full trial enrollment in mid-2025. With that, I'll now turn it over to Christoph to get to the heart of our call today, data results from the completed phase 2 study in chronic neutropenia. Christoph?
Thanks so much, Paula, and hello to everyone on the call. Let me begin with a quick refresher on chronic neutropenia. The hallmark of chronic neutropenia is abnormally low levels of circulating neutrophils that are the defense mechanism our body uses to fight pathogens. As we've presented previously, there is a well-established correlation between circulating levels of neutrophils, or absolute neutrophil count, ANC, and the risk of frequent and/or serious infections. Note that the lower limit of normal for a neutropenia diagnosis is 1,500 cells per microliter. Below that, grades are divided based on the infection risk in increments of 500 cells per microliter, from mild to moderate to severe. As you can see, severely neutropenic patients have ANC of less than 500, and this corresponds to the highest risk of infection. To reduce the risk of infection in these patients, it is critical to move them from the most severe grade towards less severe grade to achieve as normal a level of neutrophil as possible. Therefore, an increase of 500 cells per microliter is an important clinical practice objective. Through its mechanism of action, XFOR has been shown to do precisely that, increase the number of circulating neutrophils. The ability is clearly stated in the XFOR label from the FDA approval in WHIM syndrome based on results from a pivotal phase 3 trial. These results demonstrated that XFOR sustainably raised ANC over the 52 weeks of the trial and consequently reduced the analyzed rate of infection, along with the duration and severity of infections in trial participants. This infection benefit was achieved through an increase in mean ANC of more than 500 cells per microliter. These results and the WHIM approval in the US provide confidence in our plan to potentially bring XFOR to those with other chronic neutropenic conditions where significant unmet needs remain. The gap in addressing these unmet needs results from the significant lack of innovation in the field. The only currently available therapy is GCSF, which was approved almost 30 years ago. It's an injectable drug that is associated with dose-dependent and often treatment-limiting side effects, as well as long-term risks of certain malignancies. As you've heard in our previous investor presentations, CN patients and the physicians that treat them are dissatisfied with G-CSF and its side effects, especially fatigue and bone and muscle pain. Unfortunately, these side effects are often dose-dependent and can limit the use of G-CSF or prevent its use at a fully effective dose. We believe there is an urgent need for a new innovation like mavorixafor, which data now suggests could be used, if approved, as a monotherapy or in combination with G-CSF, while also allowing for reduced doses of G-CSF to limit side effects and long-term cancer risk. Our market research indicates there are approximately 50,000 people diagnosed with CN in the U.S. Of this population, it's estimated that about 15,000 have high unmet needs, including recurrent infection, despite the available standard of care. Our phase 3 FORWARD trial focuses on this population with the highest unmet needs, enrolling adolescents and adults with ANC below 1,500 cells per microliter and a history of recurrent and/or serious infections. To meet the multiple needs of this population, we envision two potential opportunities for oral once-daily mavorixafor within this market. One, it could be used as a monotherapy in newly diagnosed CN patients or to replace G-CSF treatment. And two, in combination with G-CSF, enabling a meaningful reduction in G-CSF dosing, lessening pain, discomfort, and long-term risk of malignancy. Our phase 2 CN study set out to evaluate those opportunities, and as you will see in just a minute, we believe the data from this study supports the potential for mavorixafor to transform the chronic care of this neutropenic population. As shown here, the main goal of the phase 2 study were to confirm the durability of the phase 1b results that showed 100% response to a single dose of oral mavorixafor plus or minus G-CSF across multiple CN types. We also aim to assess the long-term safety and tolerability of mavorixafor use with and without G-CSF in people with CN. In addition, we wanted to explore the feasibility and the willingness of physicians to safely reduce G-CSF with mavorixafor, and a key goal was also to inform the design of our phase 3 pivotal trial, and in doing so, de-risk the trial. The phase 2 study was a 6-month study with monthly visits and comprehensive assessments at baseline and months 1, 3, and 6. Two quick notes on the design of this study. First, since the neutrophil life cycle is about 10 to 14 days, we believe that a study of this length provides a good indication of the bone marrow's long-term ability for stable neutrophil production. And second, modifications to G-CSF dosing were permitted in the combination group following participants' month 2 visit, so the first assessment of those on an adjusted dose would have been at the month 3 visit. These adjustments were decided between physicians and their patients and were not protocol mandated. Let's start by looking at the final participant disposition in the study. A population typical of those with chronic neutropenia and high unmet needs. We enrolled a total of 23 participants in the 6-month single-arm open-label study with a mix of idiopathic and congenital CN, and even a few with cyclic presentation. There was a typical gender distribution in the study and a mean age of 34 years. We also note here that there was a good mix of genetic backgrounds with the congenital group, also representative of what you might see across a full CN population. We analyzed the data in two groups, mavorixafor monotherapy, comprising 10 participants, and the mavorixafor plus G-CSF group with 13 participants, 9 of whom ended up having their G-CSF dose reduced in the study. In addition, we evaluated neutrophil functionality pre- and post-treatment in a sub-study population of 9 evaluable participants and compare those results to a group of healthy individuals. To address the unmet needs existing with current chronic CN therapy, our objective is to sustainably increase circulating functional neutrophils using oral mavorixafor. Thus, the results presented today are focused on answering the following 4 questions. 1. In the monotherapy group, did mavorixafor increase and durably sustain absolute neutrophil count at clinically meaningful levels? 2. In the combination group, were physicians willing and able to adjust their patient's G-CSF dose when adding mavorixafor? 3. Could G-CSF be reduced while maintaining clinically meaningful ANC levels? And 4. Are the neutrophils that are mobilized into the peripheral blood by mavorixafor functional throughout the 6-month study? First, let's look at the monotherapy results. Consistent with our data presentation in June, mavorixafor durably and meaningfully increased mean ANC, bringing levels into normal range at month 3 and month 6. As an oral once-daily treatment delivering this level of ANC increase, you can begin to appreciate the potential for mavorixafor as monotherapy. In addition, mavorixafor also durably and meaningfully increased mean ANC level in the severe CN study participants. Those starting with levels below 500 cells per microliter had more than 2-fold increase in mean ANC versus baseline throughout 6 months of mavorixafor treatment. As a reminder, such an increase in ANC in our phase 3 WHIM trial led to a decrease of 60% in infection rates. So this similar increase in ANC here is not only clinically meaningful, but also helps increase our confidence in achieving a positive infection rate readout for this monotherapy population in the ongoing pivotal phase 3 trial in CN. Let's move now to the combination results. In this group, we started with 13 participants entering the study on G-CSF at baseline. 1 dropped out almost immediately, so we had 12 eligible to have their G-CSF dose reduced following their month 2 visit. Notably, clinicians chose to reduce the G-CSF in 9 of these 12, with 3 being fully taken off G-CSF prior to their 6-month visit, finishing the study on mavorixafor monotherapy. Thinking back to the market opportunity and unmet needs slides, you can now see why we're so excited about these data. This is the first look at how physicians could potentially use mavorixafor in their CN patients who are already on G-CSF should it gain approval. Today, we're not presenting the data from the 3 participants who remained on stable dose G-CSF, but we know that the final data set from this group was consistent with the results we shared this past June, showing robust and sustained increases in mean ANC from baseline levels. Let's look now only at participants who were dose adjusted. As shown here, the G-CSF reductions were quite substantial, and mean ANCs were able to be maintained at normal levels throughout the study. At month 3, when 8 of the 9 had been adjusted, physicians were comfortable lowering G-CSF by 52% on average. This average increased to 70% reductions by month 6, with 3 of the 9 adjusted dose participants taken completely off G-CSF. Anecdotally, physicians and academic experts have told us that they believe a 25% reduction in G-CSF would be clinically meaningful to their patients. So seeing this level of G-CSF reduction reinforces the potential benefits of mavorixafor and role in the treatment of CN. We now turn our attention to assessing the functionality of the increased levels of circulating neutrophil in a phase 2 sub-study. As mentioned, the purpose of this sub-study was to assess the percentage of functional neutrophils in people with CN, including those with congenital genetic variations associated with neutrophil maturation arrest. We used two well-accepted functionality assays, the phagocytosis assay, which assesses the neutrophil's ability to engulf pathogens, and an assay for ROS production. ROS is reactive oxygen species, or the ability to damage or kill pathogens that have been engulfed. These neutrophil function studies were conducted on a sample from 9 eligible participants with a mix of CN types and monotherapy and combination use. We also recruited 5 healthy individuals to provide a benchmark for comparison. For simplicity today, we are only showing the phagocytosis data, but we note that the ROS assay results closely mirror the phagocytosis results. While participants with chronic neutropenia have fewer neutrophils than healthy donors, you can see here that those neutrophils that do make it into the circulation are in fact functional, with a similar percent of functionality between the neutropenic and healthy donor population. Note that the bars here represent the percentage of neutrophils that demonstrated phagocytosis after challenge with opsonized bacteria. We've also broken out the congenital CN subset to see if genetic variants affect functionality differently than in the other groups. Looking now at full results, we see that the percentage of functional neutrophils in the healthy donors, the full group of participants, and the congenital CN subset are essentially the same following 6 months of mavorixafor therapy. And note, while we are showing you here the data for phagocytic function, we generated very similar data on the percentage of neutrophils generating ROS after bacterial challenge. Recall in our 52-week WIND phase 3 clinical trial, we saw that an increase in circulating mature neutrophils corresponded with a 60% reduction in the annualized infection rate, as well as reduction in the severity and duration of infection. By increasing the number of functional circulating neutrophils, we believe that mavorixafor treatment could correspondingly reduce the infection rate in a larger chronic neutropenia population. And we are hopeful to see similar results in our ongoing registrational phase 3 chronic neutropenia trial. Mavorixafor with or without concurrent G-CSF therapy was generally well tolerated throughout the study. The overall safety profile was consistent with previous studies, and the most frequent treatment-related, treatment-emergent AEs were GI-related, nausea and diarrhea, which were mild to moderate and typically resolved over time. As we noted in our June presentation, we did see 3 discontinuations in the study. These occurred early in the execution of the study and after implementing an education program on possible GI side effects. There were no further discontinuations. Rest assured, we have incorporated this education into the phase 3 CN trial. In summary, we could not be more pleased with the results of this phase 2 study, having met all of the goals we set forth. We have shown that mavorixafor can durably and meaningfully increase mean ANC as a monotherapy, even in the most severe CN population, and can do likewise when dosed in combination with G-CSF. We show that mavorixafor enables clinicians and their patients to meaningfully lower the use of G-CSF while maintaining mean ANC at normal levels. Critically, we show that mavorixafor increased total counts of functional circulating neutrophils even in the most difficult to treat individuals with CN. And importantly, we demonstrated that mavorixafor was generally well tolerated on its own and in combination with G-CSF. These results give us great confidence in the chance of success of our ongoing phase 3 trial and mavorixafor's potential to reduce infection risk. They also provide hope that mavorixafor could be the innovation that the CN community is looking for. As you've heard previously, through our interviews with physicians and CN patients, what they need and want is a well-tolerated oral option, one that can enable them to safely reduce the use of injectable G-CSF. And so far, our results have shown that mavorixafor fits that profile well as either a monotherapy or in combination use. I'll now turn it back to Paula to conclude. Paula?
Thanks so much, Christophe. Great job sharing our exciting update on mavorixafor's potential in CN. To conclude, over the last 6 months, we have made significant progress as a company in bringing innovation to the immunodeficiency community. By gaining approval for and launching XOLREMDI in the U.S. for WHIM syndrome, and by exploring additional geographies for mavorixafor's approval and distribution in WHIM, we are seeking to maximize the global opportunity to help those with WHIM. And by completing and reporting out these positive phase 2 results and initiating our pivotal phase 3 trial of mavorixafor in those with chronic neutropenia, we are seeking to further expand the market opportunity for mavorixafor and have a positive impact on a much larger potential patient population. In terms of our financial results, I direct you to our press release from this morning and our 10-Q to be issued shortly. We ended the third quarter of 2024 with cash and equivalents of almost $136 million. We continue to believe this gives us the runway into late 2025, and note that this does not include the expected ramp-up of sales of XOLREMDI throughout next year. As always, we thank you for your continued support and would now be happy to take your questions. Operator?
Thank you. At this time, if you would like to ask a question, please press the star and 1 on your telephone keypad. You may withdraw your question at any time by pressing star 2. Once again, to ask a question, please press the star and 1 on your telephone keypad. I will take our first question from Kristen Kluska with Cantor. Please go ahead. Your line is open.
Hi, good morning, everybody. Congrats on these new great data you shared with us. Really appreciate it. So questions that I have are related to the new data today. So I wanted to ask what the biggest criteria were in the trial that made the physicians comfortable to reduce and lower the G-CSF usage. And while I recognize it wasn't a formal endpoint measured, I'm curious if the doctors had any anecdotes to share about any early signals of reduced pain or better convenience benefits for patients.
Yeah, Kristen, thank you for the question. I'll start, and then Christophe will add some color. So, the important thing about the phase 2 study is we really put the choice in the physicians' and patients' hands. So there's a wide variety of patients. They have real-world experience with them. So really what gave them comfort to adjust G-CSF dosing was the high and robust ANC responses due to the addition of mavorixafor. That is confidence in data, and of course they acted on that. But Christophe, why don't you give some more color on that?
Sure. Yes. No, we are really happy with the results and the choice that 75% of those physicians with their patients have decided to decrease G-CSF. This is a really high percentage of those decisions without any mandates from the protocol or any prescriptions that we ask them to do this. With regard to the choice to do it is, as mentioned, a choice between patients and the physician. And it's a balance between the level of ANC, as Paula just mentioned, and so they were very comfortable with the level of ANC and the clinical manifestations that these patients were having. So we could not identify a specific pattern, but this is really what drives most of those decisions.
Okay, thanks. And then any early anecdotes, or it's a little too early to make that assessment?
I think it's early to make that assessment. And of course, what we're most pleased with is the broad applicability, the broad reduction in G-CSF across the 75% of patients, and even some of them completely coming off. So certainly a lot more study to be done, Kristen, but thanks for the highlights.
Yeah, and if I may ask one more question then, can you talk a little bit more in terms of what lower G-CSF usage actually means? Is it that physicians are assessing every other day for usage? Is it the amount of G-CSF that's administered is lowered? And then do we have a general sense to give us what correlation of G-CSF usage could ultimately truly lead to alleviating some of the dangerous side effects and the inconvenience?
Yeah, so we'll take the second part first. So what we've been reporting on from patients and physicians in some of our early research is that about 25% reduction either in dose and or frequency seems to be the threshold for something that's clinically meaningful for them. And obviously the lower the better in terms of the short and long-term toxicities experienced by dosing. And then, just in terms of percent reductions, as always based off of patient's entry criteria. The challenge with G-CSF is the heterogeneity of each patient is different, so we treated them as such as clinicians do. So that reduction reflects their entry, changes in their entry baseline levels.
Thanks again and congratulations.
Thank you.
Thank you. Our next question comes from Ted Tenthoff with Piper Sandler. Please go ahead. Your line is open.
Great. Thank you very much. Good morning, everyone. And congrats on the phase 2 data. I had one quick follow-up question. When it comes to the phase 3 trial, and I'm wondering, is there a way or are you looking at benefits, kind of following up on the last question, from G-CSF reduction? Is there any way to capture those in terms of improved tolerability, improved safety? And then I have a quick one on XOLREMDI. Thanks.
Sure, I'll turn that over to Christophe.
Yes. No, so the phase 3 trial is designed as a registration approval trial as per the FDA requirements. So we will be looking at those aspects as a follow-on study to that phase 3 trial.
Great. Okay. Yep, that makes a lot of sense. And also, I have to imagine it might take some time for some of those to emerge, but yeah, that's great. And then when it comes to XOLREMDI, you guys announced a new initiative. Can you just provide a little bit more detail around that in terms of reaching out to the patients?
Sure, Ted, just to clarify a little bit more color on our sort of patient ambassador efforts.
Yes, thank you.
Sure. Mark?
Yeah, thanks, Ted. And you're right. We're laser focused right now on raising disease awareness and driving screening for WHIM and presenting XOLREMDI as a treatment that has proven to work in WHIM. So we've just launched a new patient-directed campaign. This is a combination of digital campaigns with websites as well as hard copy material that we can provide to patients. And we think this will help to educate patients and encourage them to go and have a discussion with their physician about WHIM syndrome.
Great. Excellent. Well, thank you very much. Appreciate the time.
Thanks so much, Ted.
Thank you. Our next question comes from RK with HCW. Please go ahead. Your line is open.
Thank you. Good morning, Paula and team. I just want to focus a little bit on XOLREMDI and the uptake in the market. Can you educate us on how many patients are on the drug and what is the commercialization setup right now? And I'm just trying to understand, how long do you think it would take for the drug to be adopted, in a decent fashion?
Thanks, RK. We'll just turn it over to Mark.
Yeah, thanks, RK. As I mentioned, we are right now laser focused on raising disease awareness and driving that screening for WHIM, encouraging physicians to look for WHIM patients in their practice, helping them recognize these patients in their practice. Up to now, there's been no approved treatment for this disease. So everything starts with raising disease awareness and encouraging physicians to identify the patients in their practice. And then we have a discussion with them about XOLREMDI, and we share the data that shows XOLREMDI works. So that's really where we are now, and it's building the foundation for our ramping up into 2025, and we'll be able to share more details with you then.
Thank you. Thanks for taking my question.
Thank you. Our next question comes from Stephen Wiley with Stifel. Please go ahead. Your line is open.
Yeah, good morning. Thanks for taking the questions. Maybe one on the data, one on XOLREMDI. So I guess on the data side, was there any correlation between CN subtype and the rapidity and magnitude of GCSF dose reduction? And then I guess, was there also any correlation between the GCSF dose these patients were receiving at baseline and then the magnitude of the dose reduction they were able to achieve?
Sure, I'll start and then Christophe will provide more color. But in terms of the CN subtype, the good news is the drug seems to be robustly and consistently working. We did break out the severity of those patients because those are the toughest to treat, and we saw a robust response there. But in terms of CIN or congenital, we're seeing a very consistent effect. In terms of GCSF % reduction from their baseline, again, we're not really seeing any variability in response based on their GCSF profiles. But I know Christophe has had a lot of, data analysis, and Christophe can provide some more color there.
Yes, no, thank you. So the CN phase 2, the sample size and the number of patients in that study is too small to start drawing directions where we're going with the different type of patients. So we know that all of the type of patients that were included in the phase 2 study decreased any type of those patients, decreased GCSF and chose to decrease GCSF voluntarily. That we know for sure. The phase 3 trial will help us in having that bigger picture of how those different those different subtypes, the congenital, the different mutations in the congenital, etc. All this will be analyzed in the phase 3 study with much more detail.
Okay. And then on XOLREMDI, I know you've hit 100% of your target accounts. I think you also mentioned 60% of HCPs have increased their screening. But I guess, do you have a sense of how many of these HCPs that you're targeting are indeed actively screening? Just wondering, I guess, how low of a base you're starting at here and what kind of runway you might have. Thanks.
Yeah, so again, the way we identify physicians that we think have a WHIM patient in their practice is really using claims analysis and our market research. And these are the physicians that we're going out to really make them aware of WHIM syndrome and encourage them to start screening for the patients in their practice. And this is really where we're getting the most engagement at this point. And the other thing that we're really trying to do is reach the physicians where they are. And so we've really doubled down on our conference presence this year. As you know, these physicians come from different specialties, so we mainly focus on hematology and immunology and try to really meet those physicians where they are.
All right. Thanks for taking the questions.
Thank you. Our next question comes from David Bouts with Zacks. Please go ahead. Your line is open.
Hey, good morning, everyone. For XOLREMDI, I'm curious if you will be able to provide any type of sales guidance numbers moving into 2025. And then for the new phase 2 data this morning, I realize the numbers are small, but was there any data collected on infections, number of infections or types, and then particularly for those patients where GCSF was reduced?
Sure. So sales guidance for 2025, we're not yet providing that. We certainly, Mark mentioned, will provide some more robust insights into our commercial launch as we head into the year. In terms of infection data, I mean, as you can appreciate, this is a phase 2 study. It was not designed to assess infection rates, no randomized comparator arm, no history collected. Importantly, though, we'd really like to remind everybody, in this population, ANC is proven to correlate with infection risk. That is based on the GCSF clinical trials and everyday use. So we're extremely pleased with the fact that our ANCs are robustly increasing and the neutrophils are functional. We've actually gone one step further beyond the data with GCSF in our own study. So we really hope we can remind everybody, as it relates to infections, of course, that was part of our safety database, which will be published at a future point. But really, we're incredibly excited about the totality of evidence and are locked and loaded for the phase 3.
Okay, great. Thanks for taking the questions.
Thank you. And this concludes our Q&A session. I will now turn the call over to Paula Ragan for closing comments.
Well, thank you so much for joining us on the call today. As you can appreciate, we are very excited about these data, and we are on track to deliver value. So from all of us at XFOR, stay classy and have a great day.
And this does conclude today's program. Thank you for your participation. You may disconnect at any time.